Brucellosis in sheep and goats and prevention and control. Brucellosis predominantly manifests itself as abortions. The only thing that is very clearly visible to anybody is abortions. Once they get abortions, people will start talking about brucellosis. Unless otherwise there are no abortions, people usually they don't know much about it. But my advice is that even before the abortions takes place, we need to have a clear-cut idea of what exactly brucellosis is or what exactly other meteorological agents that causes abortions are. We need to have a kind of structured approach towards identification of brucellosis or any other abortifacient organisms that is usually present in the farm, because they are perpetual in nature. The moment is being introduced into the farm, they keep thriving in the farm, they keep thriving in the herd, leading to one to another, spreading between them through horizontally or vertically, causing the problem, and that persists in a particular form. So to get rid of that is very difficult unless otherwise we take certain measures. Coming to what is abortions, as we know, abortion is an expulsion of a fetus before the end of a normal term of pregnancy. If the fetus is being expelled exactly at the term of pregnancy, we call it as stillbirth. But if it is well before that, we call it as abortion. It can occur at any trimester, first trimester, second trimester, or third trimester. So literally, it is like, expulsion of fetus before the end of a normal term of pregnancy. And infectious abortions can be infectious, can be non-infectious. Because infectious reasons are the most prominent ones. Sometimes we sideline the non-infectious ones also. Non-infectious ones can be anything. It can be nutritional, it can be toxic, it can be purely managemental. So many reasons can be attributed to that one. The non-infectious causes, like your pregnancy toxemia, it is purely managemental, it is purely physiological, that can lead to the abortions. As a thumb rule, most of the abortions, they occur at last trimester of the pregnancy, but there are certain exceptions of that, which we'll be discussing in the due course of the slides while I am presenting it. But usually as a thumb rule, most of the abortions occur at the last trimester, or stillbirth occurs in case of infectious abortions or infectious causes. So why abortion is important? Because abortion, we'll be losing a very valuable crop, that is one thing. And then we need to wait for another complete term following the interkidding or interlambing interval, then again breed the animal, and again the animal has to be conceived, and then it goes for the again kidding. So all this cost, the input cost that we are giving, the labor, the many opportunity cost that we are conquering because of that one, that leads to a huge economic losses. It's been estimated that there is a loss of around 2,121 rupees per goat annually if there is an abortion that occurs. So this is the importance of that. And then based on the zero prevalence, it was estimated that an annual loss of rupees 9,212 crore per annum is being lost on all the livestock species, including all the domestic livestock, including the pig also. So that because of the brucellosis, only because of brucellosis, not just abortion, only because of brucellosis. So besides abortion, there are also other manifestations which are equally important. Many times we sideline that. One of the most important things is that because it's being a chronic infection, it persists inside the animal, it can lead to infertility. Infertility means we need to totally remove the animal from the herd. We need to change the animal. So whatever the cost that we've incurred, that needs to be again taken care of. That leads to another huge economic losses. And also we lose a valuable kid or lamb that we get out of that particular animal that we maintained over the period of time. It can lead to metritis, uterine infections, progressive uterine infections, then reduce the productivity rate. Then there is a prolonged interconception or the lambing kidding interval, infertility, the veterinary cost. And also because of stress, the animal leads to milk production and also the milk that cannot be consumed because of the zoonotic reasons. And also the early reduction of potential use and dose. Otherwise, the quality germplasm that we identify, the best of the breed, suppose some person is introducing into the herd. The breed may be very costlier and there may be more liking towards a particular breed in the particular area. If you are losing that costly, valuable germplasm, and again that adds to the pose. So again the same thing, what is abortion and what is stillbirth? The one that we get exactly delivered on the day after the completion of the gestation period is called as stillbirth. Before the completion of the gestation period is called as abortion. So there are many reasons for that. Mostly this abortion, stillbirth, they can be due to infectious causes. Like what we get in brucellosis, we get abortions mostly, and sometimes around 20 to 30% of the cases we also get stillbirth. Same is the case with chlamydia, same is the case with your coxella. Sometimes the stillbirth animal didn't get expelled, it actually retains inside the uterus in utero, leading to mummification. We even found cases of mummification also in brucellosis. Even in coxella, where coxella, although we don't get in northern India, but in southern peninsular India, especially in Tamil Nadu, we used to have a lot of cases coming from there. They used to send samples to us. we found that a lot of mummification cases occur in Coxella also. When we look at the serological based studies, we say that Brucella is there around 5 to 20%, whatever we call it. Again, changes from various regions, states, districts. So coming to the roots of infection and pathogenesis. So if there is an infectious cause of abortion, how the animal picks up the infection and what it leads to, and how the animal leads to the abortions and infertility and all other whatever the reasons that we get because that we find in infertility. Usually it is hematogenous spread because animal, it acquires infection horizontally. Mostly it is through coitus, it is through oral, it is through mucosal region. So it actually goes into the blood circulation, where in the blood circulation it reaches the uterus. If the animal is not pregnant, so it may be present in some other places like your joints, it may be present in spleen, it may be present in any organ. Suppose the animal conceives and then slowly the animal will be having a developing fetus. As developing fetus attracts the pathogen. Fetus is having very abundance of tissue that they're multiplying, that it can able to support and contains rich nutrients. And also it's having a very highly enclosed and highly supported system where it's totally driven away from the maternal immune system, where the fetal immune system is totally different. So looking into this opportunity, the pathogen can easily spread into cross the barrier, the placental barrier, get into the fetus and can be able to lead to abortion. This is hematogenous spread. And there is one more thing called ascending infection. Ascending infection means maybe through the genital tract. Through genital tract, it goes, establishes the infection and causes the abortions. How exactly abortion is caused? Abortion occurs because this particular pathogen multiplies in the placental area. When it multiplies, it leads to placentitis. When there is a placentitis, then there will be a problem in the exchange of nutrients from the maternal side to the fetal side. When this occurs, both the nutrients and also the oxygen supply, both will be affected. If the both are affected, then there will be a progressive infection or progressive loss of development of the fetal development occurs, and that leads to propelling of the fetus, expelling of fetus away from the maternal side. So one needs to understand that whatever abortions occur, it is not at a nick of the moment. It is actually progressive. The animal carries the particular infection, progresses, it slowly infects, it leads to complete curve of nutrient flow into the developing embryo, and also cut off supply of the oxygen is completely cut off, wherein that leads to hypoxia in the fetal development. Hypoxia leads to reduced respiratory rate or reduced oxygen transport and exchange, and that leads to slowly the death of the fetus and ultimately it gets expelled out. So fetal hypoxia is the most important thing. The fetal hypoxia occurs from the maternal hypoxia, the maternal circulatory system failure, and there is an interference in the oxygen transfer that leads to placentitis and also premature placental separation. So fetal compensatory mechanism then starts. That means it shunts the blood to the vital organs to maintain the normal oxygen levels, wherein the fetal respiration increases. And then it in an attempt to compensate with hypoxia, and then there will be aspiration of certain amniotic fluid into the fetus, and all these things occurs. As you can see in this particular photo, wherein you will find the cotyledons, they are having they are actually showing cotyledonitis, showing a focal kind of necrosis in the cotyledons. So that leads to placentitis, and then the supply of oxygen is being cut, and then we can also see the congested uterus that is uninvoluted, and you will find the typical edematous uterus where a new abortion occurs. Suppose we need to have a complete clinical history. We need to ask about when they have bred the animal, whether they brought any new bugs or rams into the farm, and whether they have been tested for brucella, tested for any pathogenic agent, whether they are vaccinated recently, because vaccination of certain live attenuated vaccines can also lead to idiosyncratic reaction and then leading to abortions. They can occur because live virus can also lead to hyperthermia or leading to abortions. They can also induce abortions. So it's very important that complete anamnestic details need to be taken from the particular farm to know about how exactly the abortions has occurred, whether they did any change of feed, whether they put in a new grazing area, whether they introduced some other animal along with the sheep or goat, and whether there are any wildlife involvement, whether there is a wildlife domestic animal conflict is there, like what we find in many free range areas. This particular problem is there. But intensification we are going for intensification, there may be certain toxic principles, there may be certain deficiency of certain minerals may be there, because we don't provide many times salts and all. That is also very essential. Salt is not important for the production. Salt is very important for the reproduction and also for essential maintenance of homeostasis and physiological balances. So that needs to be provided properly. There are certain area-specific mineral mixtures are being developed that needs to be incorporated in the feed. Like for suppose if you take the state of Telangana, so what are all the nutrients which are abundant in the particular area? We need to analyze the soil, we need to analyze the water, we need to analyze the plants, or everything need to be analyzed. And also we need to analyze the blood of the animal. We need to identify what are all the important micronutrients which are present, and accordingly we can deal the non-infectious causes of abortion. Coming to the infectious causes of abortions, we take all the biosecurity measures. Whether there is any breach in the biosecurity measures recently in the form, whether new animals have been introduced, whether quarantine protocol has not been followed or not followed, and what is the time of abortion, whether you've been giving them any different kind of stuffs to the animal, like introduction of new RO water and some kind of different type of feed you are providing, any change in the feed. So whether there is a change in the environmental condition recently, all this kind of history need to be completely taken to assess, especially in the farm history in a herd. What are the type of samples that need to be collected? If it is aborted fetus, one needs to take proper precaution. We need to wear proper gears, especially one need to wear the goggles, we need to wear masks, we need to wear gumboots, we need to wear gloves. So once we collect the sample, you can collect it from the genital tract, you can collect it from the safely. If you are collecting from the aborted fetus, that needs to be opened in a separate place where you can collect the sample, and that needs to be properly buried following all the biosecurity measures. So you can collect the samples. Or if you're screening the animals, suppose you want to screen the animal, you can collect the blood, you can collect the genital swabs. Suppose if there is any shedding of this particular pathogens, they can be a possible threat to other animals also. So all of the animal needs to be checked properly. So sampling is very vital. And again, how we need to send the samples, it depends upon the place where you are studying, the distance of the place of the lab which is located. Suppose the lab is very nearby, you can simply collect in a small ice pack and you can send it. Suppose if you are sending to long distances, you definitely require dry iced, a robust, the one that supports a completely favorable environment for preservation of your samples. So all these things are essential. We are using the swab-based techniques. As you can see in this particular picture, you can collect the swabs. Even if you don't have this kind of fancy swabs just supplied by any bigger chemical company, you can simply use earbuds. You can use autoclaved earbuds. So collect using the earbuds, by wearing a gloves, you can collect the samples from the genital environment, and then you can put it in a Ziploc cover. You can put it in an envelope and you can send it to the laboratory. Like we are receiving like that only. There are no eyes required. Simply you need to put it in the envelope properly sealed. So that because there may be some infection, we need to seal it properly. That kind of responsibility we need to take. Certain cards, like population cards are there, where if you cannot able to send the blood samples and serum, simply you can put a drop of blood into an area of paper. Small paper is there. So they are called as population cards. You can put it in that one where you can put their ready-made envelope is being given, and you can simply send it by post. We used to punch that particular area where the blood has been dried. So we used to elute it. So after eluting, we used it for doing the ELISA technique. So these kind of techniques are already being followed all over the nation. Or you can go for lateral flow assays. Lateral flow assays is that we need to take a proper decision or interpretation that is highly important. We may get a negative or positive, but so what? After getting a positive, so what kind of precautions we need to take and how we need to interpret? That's highly very, very important. Because certain tests, they cannot directly say that this is Brucella and this is what you need to do. We need to do a repeated test. Repeated test is very, very important. We need to repeat the test again. Then we need to go for another set of tests, because simply getting a positivity in a particular test need not be a true positivity. So we need to go for, exactly, we need to measure it. We need to detect it again by using another test, comparing test. So this is very important in Brucella, because a decision needs to be taken supported by at least more than one test, so that our decision can be better. Brucellosis, as we know, is also highly zoonotic disease, is highly infected human beings, especially the pathogen that is Brucella zoonotis is the most important pathogen for human beings, and is highly infected human beings and highly hazardous to human beings. So we need to take all proper gear, all proper precautions. So it also, besides abortion, it also causes infertility, metritis, orchitis, and even male also infertility is very common, where the brucellosis is there in a particular endemicity is there in a particular area. So the bucks and the rams needs to be changed regularly, because of course for breeding reasons, of course for genetic reasons, people say that we need to change the bucks and the rams regularly, and also we need to test them properly. And before testing them, we need to quarantine them for at least for a few period for 21 to 28 days. Then once you're pretty sure, after double testing by using companion tests, that this particular animal is free of brucella, then we can introduce into the other herd by following all the biosecurity measures like vaccination, deworming, all of the things needs to be followed. So this is usually done in an organized form. But whereas in a herd, where it has been unorganized, where the farmer is maintaining, this is highly difficult. Farmer needs to think how to reduce the abortion if he is having any abortion in the particular unorganized herd. So he can contact the local veterinary dispensary, local veterinary doctor. We can get the sample, we can get it sampled. Or CRG, we are working towards how to actually reduce the transmission, horizontal transmission among the field flocks, because field flocks is very difficult to test them. It's very difficult to reach them. And even if it is easier, even if you could able to reach them, it is very difficult to control those animals, moving from one place to another. Because there are many kind of, herds are there. Moving herds are there. Stationary herds are there. Especially in the case of sheep. Especially in northern India, you will find people migrating from Gujarat to UP to Gujarat, people coming from Himachal to Haryana back to Himachal. So these kind of, migratory flocks are there. So these kind of migratory flocks, they don't have any control at all, unless otherwise we could able to come up with a strategy totally different from the diagnostic testing, rather reducing the horizontal transmission of brucella from one animal to other animal. So we have made a formulation called as Bruclear. So this particular Bruclear is a herbal-based formulation, wherein this particular formulation being fed to animals for 15 days, it can reduce the horizontal transmission of, although it cannot totally remove the brucella from the particular animal, but it can completely reduce the shedding, so that you can warn the farmer to not at least breed this particular animal. Even the animals, other healthy animals which are being kept along with that, they will not be under risk. Coming to the various species of brucella, Brucella melitensis is having the highest zoonotic potential that affects both goat and sheep. Brucella ovis is also in the sheep, but the first three melitensis, abortus, and suis, they are having a very high zoonotic potential, whereas Brucella ovis is non-zoonotic in nature, and you can handle them in the biosafety level 2 laboratory itself. Whereas the first 4, it requires biosafety level 3 laboratory. Biosafety level 3 biosafety cabinets are required for handling these pathogens. But the most important thing I want to say that Brucella, as commonly what we think is that Brucella spreads only by coitus or by sexual intromission. It is not only by that. The most common route is oral route, because the contaminated water, the contaminated pasture, the contaminated feed, these are the predominant cause of brucellosis in the small ruminants. It's not only by breeding. Breeding is one way, but keeping a brucella infected animal along with the healthy animals itself can be dangerous to the other healthy animals. Even if it is not bred or not bred, that is not important. That infection cycle is always going on, because the route of infection is by oral route. So oral route leads to the most major causes of infection in the domestic animals. So we need to check each and every animal before breeding them. So even if we are having some 7 adult female or some 10 adult male, each of them needs to be monitored properly before you're going for breeding. So many times we will be looking for Brucella melitensis in sheep and goat, because it is a predominant pathogen in sheep and goat that causes abortion. Brucella abortus can also cause abortion in sheep and goat, but it is very, very less. But it can be carried in the Brucella abortus can be carried in the small ruminants. Similarly, the Brucella melitensis can be carried in cattle, buffalo. It need not cause abortion, but it can be present in the cattle and buffaloes. So they are interchangeable, but predominantly the abortions occur in small ruminants because of Brucella melitensis. In India, this particular disease is endemic based upon the whatever serosurveillance that we have conducted in the previous years. Coming to this particular slide by Nivedi, Shom et al., the prevalence of brucellosis in sheep in India. In the left, you are finding the table where they have studied in some 13 states, and they have studied the apparent prevalence and the true prevalence. Of course, the true prevalence is based on the specificity of the particular test. That says that it starts from around 0 to 28.6%, 0 to 30% what they found. And you side in the right side, there's a map is that is in 2020 they studied. They found that especially in Karnataka, in Gujarat, they are showing very, very high prevalence of brucellosis. Whereas in states like Andhra Pradesh, in states like Maharashtra, Uttar Pradesh, Haryana, it is showing little lesser, around 15 to 20%. Whereas in other states like Tamil Nadu, they show around 0 to 5%. So many states have not been properly tested, like probably Bihar and Arunachal Pradesh are not tested. But as you can see in this particular map, that India, we are having a widespread brucellosis prevalence is there in sheep. Coming to the brucellosis in goats, And again, we find around the true prevalence rate is around 2 to 10%, what they studied. But in 2020, they again studied the separate set of samples where they found that is around 0 to 30%, again, the case of goats also. So this is in the percentage positivity of brucellosis in sheep and goats based on the study conducted in 2020. As you can find, the apparent prevalence of brucellosis in sheep, which is shaded blue, the blue histogram, it is around 25%, highest in Karnataka, followed by Gujarat, Maharashtra, Uttar Pradesh. Probably in Karnataka, Gujarat, Maharashtra, Uttar Pradesh, Haryana, the intensification is also increasing. Slowly, it's slowly increasing. So in the states starting from Karnataka to Tamil Nadu, the intensification is slowly progressing. That's the reason why we are finding little increased incidence of brucellosis. Probably the states like Tamil Nadu or probably Telangana, they have some kind of better control strategies, or probably they have better strategies, biosecurity measures. What you find, lesser incidence of brucellosis compared to Karnataka, Gujarat, or Maharashtra. Another thing is that the zoonotic risk that we are finding in humans, the annual human brucellosis incidence per million population in the world. In India, you can find it is possibly endemic, which is shaded yellow, and also certain African countries, they are shaded yellow, where it is possibly endemic even in human beings. So we need to look into the broader picture, like why humans it is present, and what are all the causes that what are all the repercussions that we're getting in human beings because of brucellosis in small ruminants specifically, or large ruminants. The more and more we are going towards intensification, the more and more handling that we do, the more and more handling we do, the more and more the infection starts spreading from the animals to human beings. And again, the consumption of milk, especially in certain states where they consume raw milk, the raw milk consumption is highly dangerous, where people, they directly drink from the teats from the udder, or they directly they consume the raw milk. That can promote, even that can lead to tuberculosis also. So this is one of the leading causes of tuberculosis in actual human beings when they consume the raw milk, the Mycobacterium bovis, which is commonly present in the bovine milk, that can lead to tuberculosis in human beings. Same way, brucella is also chronic pathogen that also can affect the human beings. So effects of brucellosis in animals can be direct or indirect. Like visibly, you will find losses in abortion, reduce in milk production. There is an invisible thing, that is the reduced fertility, that one needs to look into. You may be getting abortions. Farmer may be thinking that this time I got an abortion, so next time I will not get. I probably provide better healthcare, and then again I'll breed the animal. But the interkidding or interlambing interval will be wrong, because of infertility, the animal may not take up the breeding. Animal may not conceive because of natural service or artificial insemination. And also the additional costs that one incur because of the abortions. So phylogenetic proximity means there is shared antigens are there. That's also one more important thing. So you'll find more false positivity, because Brucella abortus and melitensis, that can lead to false positivity, because most of the diagnostic tests currently that we're using they are abortus based. So abortus based, they can react to the O antigen, that is a lipopolysaccharide induced antibodies in the animals, that can produce a false positivity. So one needs to be very, very careful while interpreting the serological based tests. Is very important, because many times because of false positivity, we may exaggerate the results, that can lead to little more problem in the policy level decision at the particular state or the country. That one needs to be very, very careful when you're reporting brucellosis, especially in international papers. One needs to be very careful, because it affects our perspectives, economic perspectives at the national level. So my humble suggestion is that it's very important to look into other perspectives before actually coming up with the positivity for a particular test for brucellosis. The brucella symptoms, as I was explaining, that majorly that we get reproductive failure, abortion, and stillbirth in sheep and goats. Besides, we also get retention of placenta. ROP is very well there, but there are few cases where ROP is not being exhibited. That's why I say, just by looking at the type of abortion, one cannot be able to come up to conclusion that is brucella, chlamydia, or whatever it is. So most infectious abortions, they occur in the late trimester, last trimester, but abortions can occur due to infectious causes in the early trimester also. Like for example, in the case of tritrichomonas fetus, a protozoan disease that can occur in the early trimester. In case of Toxoplasma gondii, it can occur at any term of the gestation. The abortions can occur. In case of viral disease, it can occur at any term of the gestation, like blue tongue, like PPR, like FMD. They all can induce abortion at any stage of gestation. So one needs to be very careful in interpreting the abortions. So it's very important based upon the data, clinical history, and also the proper testing is very, very important to actually say that, to confirm that this is brucella, chlamydia, campylobacter, or coxiella, or whatever it is. In male, acute orchitis, epididymitis is there. When male also loses its fertility slowly, if abortion is persistent in a particular herd, as you can see in the particular picture, sheep being showing the orchid is because of Brucella melitensis infection, where there will be enlargement will be there, there will be hydrocele may be there. So this particular problem is always there. So this particular male, once we are ignoring it or overlooking it, they can be spreading the brucellosis by even just remaining in the herd. You will not breed them, just put it in the herd. There will be the smegma may be carrying, it may be a rich source of brucella and other causative agent that causes abortions that can be spreading into the environment, leading to rendering other animals also getting infected and leading to havoc in the particular herd. One needs to know that brucella usually is not present freely. It's not cell-free, because brucella goes into the thing. It enters the immunological, all kind of immunological functions that occur inside the system. When there is a macrophage activation is there, the brucella enters into the macrophage via the phagosome. So this particular phagosome is there. The phagosome needs to be killed, right? Because it contains infectious agent. So there will be lysosome will be there, that will be fusing with the phagosome, and then there will be a respiratory burst, and then there will be killing of the particular pathogen. But what happens is that brucella evades all this particular thing because it actually hoodwinks the complete immune mechanism. There are various factors other that will be explaining, in the further slides. It hoodwinks the mechanism and slowly it multiplies inside the phagosome. And then when there is a favorable time, they will be released from the phagosome, and then it infects more and more number of cells, and it is driven towards the reproductive tract, driven towards the uterine, driven towards other organs, leading to the exhibition or manifestation of the clinical symptoms like abortion or other kind of the whatever manifestations that we get. So one needs to critically look upon why exactly brucella resides in that, because brucella lacks many classical virulence factors like pili or capsule. Many things are not there, but brucella still tries to hoodwink still able to hoodwink the immune mechanism of the host. It contains various factors. So the main factor is that the lipopolysaccharide that is present in brucella is totally different. It is a non-classical LPS. The classical LPS is usually different. This non-classical LPS, it contains a lipid A, which is connected to core by amide bonds. And again, this particular lipid chain is quite longer compared to the E. coli, the one that we find in E. coli. So the E. coli is a classical gram-negative pathogen. So this particular thing is lacking, and it's present in two forms: rough and smooth form. The smooth LPS forms are highly pathogenic to human beings, very highly pathogenic, and they are very pathogenic to the primary host also. Like Brucella suis for pig, Brucella abortus for cattle and buffalo, Brucella melitensis for sheep and pig. That is their primary host, because it contains O chain LPS. Whereas there are rough mutants are there, like rough forms, they lack the O chain LPS, but they are less pathogenic or non-pathogenic to human beings, but they are highly pathogenic to their primary hosts again. I repeat again, the smooth LPS is highly dangerous for the human beings and the primary hosts, whereas rough forms, they are less infective to human beings, but they are highly infective to the primary pathogens. That's why the rough mutant forms like RB51, that's why they are promoted in US. Like RB51 was less infected to the human beings, they promoted it, but the problem was that they don't have a proper DEWA strategy, and also they may be, even though they are being used as a vaccine, they can anytime become pathogenic again. Despite the attenuation, they can cause abortions. So this is another problem that I'll explain in the coming slide what exactly happens. Brucella contains a very long chain fatty acids in its particular lipopolysaccharide, up to C28, carbon 28, compared to the E. coli, which is comparatively shorter chains. So the problem is that when you're having a longer chain of C20, longer chain of these fatty acids, it actually leads to suboptimal TLR4 activation. That means immune activation is not proper. Otherwise, what you find in E. coli, because of the shorter chain fatty acids, the proper innate immune responses are there, leading to the proper deployment of the immune cells. Whereas in the case of brucella, it is not happening because of the change in the lipopolysaccharide structure. So this is what you find. The brucella takes a very weak acid for staining, what you call as modified Zielinson staining. So this particular staining is to differentiate from the other gram-negative organisms. But if you use selective media, we can able to grow brucella. The most important thing I say, the most specific test is definitely the culture one, but culture requires biosafety level 3 biosafety cabinets and laboratories. So that's why usually we don't try to maintain brucella in the laboratory, but we try to have all the diagnostic tests, diagnostic tools like primers and all, where we can able to diagnose the brucellosis. The diagnostic challenges that we face in brucellosis in small ruminants is that the most available diagnostic reagents are from the abortus, origin of Brucella abortus. So this leads to kind of, although I don't say that it is a problem, it's a big issue. If you have very good, a capable person to interpret it, but in toto, Brucella abortus can lead to little more reactivity and little more false reactivity, and it is having a false positive reaction with the febrile nature of the animal. Suppose the animal is having some other fever, then there will be antibodies to the O antigen, that is lipopolysaccharide, that can very well cross-react with your Brucella abortus antigen, the antigen that we're using, and that can able to produce the positivity. So this particular test sometimes cannot be taken as a true positivity. So we need to go for a companion test, check again based on that. Suppose most forms they use this particular plate accreditation test or slide accreditation test. This is a very useful indicator of whether Brucella is present there or not. So I call them as a kind of indicator testing. Indicator testing means whether a particular herd is having Brucella or not. You will just get an idea, but you cannot confirm that this particular animal is suffering from Brucella. We need to go for a better testing, better technique. What I suggest is that suppose we go for SAT is a very, very low sensitive test. I say that it may be highly specific, but it is having very low sensitivity because it requires high quantity of antibodies to produce it. And again, the problem is that if there is a prozone phenomena, probably you may not get in the first tube. We need to go for a multiple number of tubes. Even if you are getting SAT positive, and again say that it is better to go for testing in paired sera samples. Test again after 21 days. You just see that whether there is an improvement in titer. If there is an improvement in titer, probably I can say that being a chronic disease Brucella, there will be an increase in the titer because there is an establishing infection is there. And again, one more problem is that whether they are IgG or IgM, that we don't know. Most probably you will be getting only IgG or total IgG plus IgM. I suggest that to know that if the particular infection is recent, then we need to go for IgM ELISA. IgM ELISA will be giving a better picture that whether these animals have been recently infected or whether these animals have been infected because of introduction of a new animal in the herd. So there are multiple perspectives are there in the interpretation of the test. That's why I say that lateral flow assay technique may not help the farmers, even if they identify the animal is positive to brucellosis or negative to brucellosis. It's always better to go with a companion testing. It's always better to send it to a place where experts are sitting and they are doing the test. They can able to interpret better along with the clinical picture provided by the farmers. So these kind of bottlenecks are there Brucella because diagnostic tests are always confusing if you don't have a clearer picture of what is the true specificity of this particular testing and what are all the other repercussions that we used to get based on the testing. Interpretation of suppose I do SAT on goat blood, 40 international units and above, that is in the third tube, more than 3 tubes in SAT, I call this a persistent infection. Suppose this particular titer may increase also when there is an infection is going on in the body, progressing infection is there. And if I am getting 40 exactly, it's an established infection. And 20 international units, usually we call it as establishing febrile illness. Because when there is a fever, even if you're vaccinating for PPR, if you collect after a week, you may be getting 1:20 titer. That's why I am telling it needs to be very carefully taken into account that what kind of antigen you're using and how many tests you're using for zeroing in towards the brucellosis in small ruminants. ELISA is a very important tool. Suppose if you are going for a population screening. Suppose if I have 500 animals, I want to do for ELISA for 500 animals in a herd. If I am having ELISA lab, I usually can able to do 1,000 animals I can do, 1,000 samples I can do in one day. If I have 2 SRF or 2 technicians, I can do 10 plates a day easily without any problem. And interpretation is quite easier because we have got proper statistical methods to project it. But ELISA, and again, I don't say it as a confirmatory tool. ELISA is a tool to know the incidence and prevalence of brucellosis in a particular geographical area. To know this particular thing, ELISA is very, very useful. Suppose if you are vaccinating, probably you can do that. You can do ELISA. But the thing is that we need to have proper DIVA strategy, differentiation of infected versus vaccinated animals. If you have a strategy, ELISA can be a very beautiful tool. But supposed to know that whether the particular area has been endemic to enzootic or there is a presence of brucellosis in a particular herd. And again, ELISA is a very excellent tool. If you are handling mass samples, a huge number of samples, ELISA is a beautiful technique. To know the recent infection, IgM ELISA will be very, very useful. Suppose if you are having a form of 100 animals, if you are introducing some 10 new bulls into the herd, so probably I can do IgM ELISA to know whether this particular bulls have contacted the infection to other animals. But it is having a very low to moderate specificity. Specificity is actually a problem. So we need to calculate the true specificity or true positivity of the particular test based upon the specificity. Usually, we get suspected to low positivity for IgG ELISA. What we have taken a structured sampling from Puducherry recently a year back. We collected from 12 clusters of coastal region of Puducherry from sheep, especially from goats. We found that most of the animals, they fall under the suspected category because ELISA cannot able to tell exactly, it can confirm it. Low positive to suspected category were more number, whereas positive category are around 20%. But this gives an idea that this particular state is having some endemicity. Using a single test, we cannot able to say. We have to go for at least one more companion test and IgM ELISA to say that what kind of picture is going on as far as brucellosis in small ruminants. So that's why we come at like antigen-based tests or molecule-based tests wherein to declare positive. Like for example, we have developed in a laboratory OM31 Tachman probe PCR. In between Brucella abortus and Brucella melitensis in OM31 gene, there are six nucleotide deletion is there. So we developed a probe that exactly binds to that particular area that is absent in Brucella abortus. That means our probe will bind only to Brucella melitensis and not to Brucella abortus, saying that this is only typically Brucella melitensis. And of course, we have got one more PCR that will detect it is brucellosis or not. In general, brucellosis, it is Brucella melitensis, or we can also detect this Brucella abortus. That give a proper picture that what kind of brucellosis is going on. It is Brucella abortus or Brucella melitensis, or in general brucellosis is there. Probably we can zero in and we can confirm it. And we can able to even know the live bacterial shedding also. If we can go for mRNA-based PCR, where the transcriptionally active live bacilli only will be expressing the OM31 gene, that will be present. If that is being shed from the animal, we can say that how much infectivity is there in that particular herd and how hazardous this particular animal is there, which is shedding the live brucella by using the mRNA-based Tachman probe PCR technique. The same thing we are following for MAP also, because in chronic diseases is a problem because they are shedding it. Because every time the animal will not shed. Sometimes the animal may not be shedding. It will be completely hoodwinking the immune mechanism, presenting somewhere lodged in the organs. And whenever there is a favorable environment, it leads to shedding in the environment, leading to hazardous risk to other animals and the human beings. So this sort of techniques are very, very essential to exactly to zero in how hazardous the positive animal is. Suppose the animal is positive, simply we refrain from the breeding. If it is not herding, if it is not being shedding the bacilli. If it is shedding the bacilli, then it is definitely a big environmental and hazardous risk for the other animals also. That is why we did all other samples like milk sample, vaginal swabs, blood. So just to know that whether this particular animal is being harboring, it is live, how much active, transcriptionally active or not. So this gives a proper picture, but it cannot be done at nationwide. But if you want to confirm a particular animal, suppose if I'm having a very high quality Jamunapari breed, they are found positive for brucellosis by SAT or by ELISA, I really want to know whether this particular animal is carrying Brucella melitensis or not. If it is carrying, it is really live or not. So all this kind of information I can able to get based on this particular testing. So this is a high specificity of Brucella melitensis, especially by using this OM31 Tachman probe. It is saying that Brucella melitensis positive control is positive, whereas Listeria, the other gram-negative organism like abortus, even the closely related Brucella abortus has been declared negative by the machine. Salmonella typhi, the another important gram-negative bacteria, E. coli, they are all being negative. So that's why they said this particular test is having very high specificity. Specificity means identifying a negative as negative is called as specificity. Identifying a positive as positive is by using a particular test is called as sensitivity. Whenever, no matter how less positive it is, by my test can able to identify it as positive, it is this particular test is having very high sensitivity. No matter if the particular animal is not carrying any infectious agent at all, if my test is detecting it as negative, that means highly specificity. If it is not carrying any infectious agent actually in true sense, if my particular test is identifying that as positive, that means it is false positive. So a test needs to be tested in all those backgrounds by using all these particular important controls to actually qualify this particular sample is positive or negative. To qualify to say that, only then I can able to say that. So we also develop other important tests like just visually if you can able to identify it as Brucella positive or not. We developed a visual lamp. So this particular apple green fluorescence is called as declared as positive, whereas this pinkish colored or amber colored what we call as negative. So this particular test was very useful earlier, but now there are less takers. And again, there's a problem with this test is that there is a no template. There's a false positivity is increasing because of NTC, the generation of NTC and contamination of naked DNA and NTCs. So this particular test we have tested in mass, a large number of samples, really very useful. Just looking into the naked eye, you can able to test whether it's positive or negative. So coming to the DIVA diagnostic test, are there any DIVA diagnostic tests for Brucella available in India? My answer will be yes, because National Institute of Animal Biotechnology, I recently read in the news, although I could not able to get much details, they developed a very low-cost diagnostic test developed by NAB, probably Dr. Grish Radhakrishnan from NAB. He has developed this particular test and they have transferred it to Ingrave Biolabs, Hyderabad. I heard that this particular diagnostic test is having a DIVA capability to differentiate between infected and vaccinated animals. Definitely, this test will be boon if it is being brought to the field level, especially for whenever you're going for eradication of this particular brucellosis by using long-term vaccination goals. Probably this particular test will be very effective. The diagnostic test versus samples tested. So that's what I explaining to you. SAT, the specificity is 75%, ELISA 90%, visual lamp 98%, OM31 Clarkman Pro 100%. Based upon the specificity, you will be calculating the true positivity of the particular test. So apparent positivity and true positivity, that will be varying depending upon the specificity of a particular test. And again, these particular things are very important. Matching, at least you need to go for more than one particular diagnostic test to declare and confirm a particular animal. Although the gold standard is always culture, but culture is definitely, it's not be possible in all the laboratories. We need to go for multiple diagnostic tests to declare an animal as positive and confirm it. In CRG, we used to have a lot of around 20% Brucella incidence, only Brucella incidence, 20%. By 2017-18, the Brucella case is almost nil. So we follow a typical biosecurity measure. We have our own SOP where we keep it quarantine, where do all the biosecurity protocols. We do Brucella testing before introduction of the animals into the herd. And every year before the start of breeding season, we test the bugs. Even we test the suspected does and the sheep also before going for eaves also, before going for breeding. And this kind of intensification of testing has led to reduction of brucellosis, where now we have zero incidence of brucellosis in, say, over a period of years. Because Brucella transmission occurs not just by sexual or coital, it is by other ways also, by secretions, by food products, by feed, by milk, and all other things. In human beings also, there's a zoonotic angle. By drinking raw milk, the human can able to contact that. And human, it will be a kind of chronic disease. There will be a perpetual transmission will be there in various organs, leading to kind of lapsing fever or intermittent fever will be there in the case of human beings. And there is when then there is a immunocompromise, definitely there will be a fever will be there in the human beings. Coming to the vaccine, the control of brucellosis is really very tricky. When I say whether Brucella vaccine is available in India or not, I'll say yes, Brucella vaccine is very well available. Whether the animals have been vaccinated or not, partially true. Animals have been vaccinated. Large ruminants vaccination is going on, especially cow food vaccination. It is very effective vaccination by using strain 19 vaccination. For sheep and goat, there is a vaccine is available called as Rev1 strain. This Rev1 strain is a very important strain. It can able to offer protection, very high protection, and is a very good vaccine. But there it is having a lot of other problems also. That is the reason why we are not following widespread Rev1 vaccination in case of small ruminants. That will be coming in the other slides also. Then there is one more vaccine called as RB51. That's called as rough mutant Brucella 51. RB51, that is between the primary host, there will be less spread of vaccine will be there. High spread of pathogen will be there. But to human beings, it is less hazardous. That's why this particular vaccine have been practiced in the Northern America. Other killed vaccine are also there, like strain 45/20, Brucella abortus, Brucella melitensis 53H38 strain are also there. But I say that Rev1 strain is the most important, highly effective strain of vaccine that is very useful in the small ruminants. So why I'm saying that if you're doing this vaccination in young replacement animals, there was a problem in controlling the brucellosis. The thing is that if you're going for high doses, it is highly effective. If you are reducing the doses, it is less effective. The problem is that higher doses can also lead to abortions, and lower doses they are not infected human beings, whereas higher doses they can be infected human beings. But higher doses they are very highly effective, especially when you are doing regularly in a particular form. But there is always a risk or hazardous of excretion of this particular Rev1 strain from the congenital mucosa, genital mucosa, oral mucosa of this particular animals that have been vaccinated with higher doses. In case of full dose, definitely there will be excretion in all the mucosal membranes. Half dose in non-replacement animals. And low dose, definitely it offered some protection, but abortions were there in animals which have been, but it is low dose is actually very, very safe for human beings, but low dose doesn't offer any good protection. Why Rev1 vaccine is not followed in India? This is a very important question. Rev1 vaccine is available, but why it is not followed? Vaccination induced abortions in pregnant animals is very common. This is one particular part of the story. Transmission of disease in humans through vaccinated animals. Definitely, this is a very big risk. And other important thing is that why in veterinary side is being discouraged is that there is a resistance of Rev1 strain for the antibiotic streptomycin. Suppose if this particular strain has been perpetuating in animals and slowly it can regain its infectivity, that means virulence. And when there is a virulence, it will not respond to the antibiotics. That means just forget about the animal. And another important problem is that this will be perpetuating. It will be slowly being spreading to other organs also, to other animals also by horizontal transmission, and to human beings also there is a zoonotic risk. And last one is it creates hindrance in serological detection of infection. Of course, DIVA strategy is important. We don't have any DIVA tool now. Suppose DIVA tool from NAB comes to the market, probably the last question will come to picture. Whereas the other three questions, vaccination induced abortions, transmission of disease in human beings, and of course this antibiotic resistance. These three are very important questions that needs to be answered if you're going for Rev1 vaccination. Other vaccine options that have been attempted previously, like Brucellosis suis, I told you, it is having a moderate zoonotic risk. They went for the attenuated strain with smooth LPS, but again, smooth LPS, I told you, it is highly infected to the human beings. Live attenuated rough Brucellosis strain like SLPS lacking, and again RB51 by using 16M strain, they did the transposon mutagenesis, and other strains are also there. Brucella DIVA vaccine in India, it is very well, I think very well been introduced. I don't know if it is available field or not. This particular vaccine is called as Brucella abortus strain 19 delta per vaccine. This is a deletion mutant vaccine. This has been developed by ICR with industry partner MS Hester Biosciences Private Limited. And this particular vaccine is having very promising results because they found that it offered protection against the virulent challenge in experimental mice and also buffalo. In buffalo, they did in National Institute of Animal Health, Bagpat, Uttar Pradesh. They found that this particular vaccine can able to have a better efficacy and also more productivity. And challenging studies produces particular favorable results. I think this vaccines are all having a DIVA compatibility because there is a strain delta per vaccine. There's a mutation of a particular gene is there. This particular gene epitope we can use as a companion test for differentiating the infected versus the vaccinated animals. Probably if this particular vaccine comes into the field, maybe we can have a field study that can able to tell that how effective this vaccine can able to perform well in the field and in the long term, and what are all the perspectives that we can look into for eradication of brucellosis in India. Suppose this vaccine works very well in the field. So it all depends upon the structured field studies required to implement it in the field. Based on many countries, they are following this particular progressive brucellosis control and eradication strategy. They have made the brucella eradication strategy by using 3 phases. Phase 1, where you can go for a mass vaccination. Again, mass vaccination means that we're having a better, high efficacy with close to 100. Actually, it's not 0%, it's 100% coverage rate, close to 100% coverage rate. We should not leave any animals. We should try to vaccinate each and every animal annually, or whatever the stipulated period we need to follow the continue the vaccination. This mass vaccination along with the DIVA strategy. The phase 2 is the combination of vaccination and culling of the seropositive animals. Culling means stamping out policy. You do vaccination, whatever positive animals you are getting, go for the stamping out policy. So this can be done for another 10 to 15 years if we follow that. Probably we can totally rule out brucella in a particular country. But another thing is that phase 3, test and slaughter. Suppose test and slaughter, as they say, that it is very effective if the prevalence is less than 1%. Suppose of 100 animals, if 1 animal is there, I can bound to lose animals. If 10 animals, 15 animals are there, I can lose those valuable germ plasm. That means other methods will come into effect. So this is a real challenging thing. We need to go for a phase 1 DIVA along with stamping out policy in the phase 2 slowly. Phase 1 has to start, maybe after 2 to 3 years, we can go for phase 2. Phase 2, we can stamp out the animals. Then slowly, if the incidence rate is reduced to 1 to 2%, we can go for a nationwide stamping out policy of brucella animals to completely eradicate that one. Of course, you can challenge this particular strategy. There may be a lot of other things are there that need to be taken care. Maybe a state-based policy may differ with the thing. The total economic losses in various ruminants, in the case of goat, it is 43.31 crore annually because of milk loss due to abortions. And again, there is one more slide you can see. There is a milk loss due to increased intercalving or interkidding or interlamping. Probably in goats interkidding, it is 63.63 crores. In case of sheep, the value of the kids or lambs is 15.4 crores. In case of goats, it is 36.1 crores. Probably if you can able to look at this particular slide, it is having a very valuable information, like what are all the, even if you bifurcate, what are all the various input costs, output costs, and what are various outputs based on the losses that we are getting incurring annually in all over India, only because of brucellosis. In goats, there's a loss of 506.19 crore per annum because of brucellosis. Loss due to milk production is 43.41 crores, and loss due to carcass weight is 298.46 crores per annum. In case of sheep, it is 155.19 crore per annum. In sheep, only because of reproductive loss, the economic loss is 97.63%. Coming to Brucella ovis, I want to skip this particular slides because Brucella ovis is not there in India. But maybe, if for the sake of knowledge, it is a risk group 2 pathogen and cause epididymitis and reduce fertility in rams. And abortion is not very highly evident. But in India, this particular pathogen is not at all reported. Maybe few reports may be there serologically, but this particular pathogen is not reported. And Brucella ovis is not present in sheep and goat. The predominant pathogen that causes abortion in sheep is Brucella militensis. Why I just want to include a few pathogens like chlamydia and all. Why to say that sometimes they may get confused with brucellosis. Even because they give a similar picture. Like brucellosis, there is a last trimester abortion is there. You will find focal necrosis of cotyledons. If you are getting the placenta, mostly retention of placenta may be there. Sometimes along with placenta, it will be there in the case of chlamydia also. Chlamydia, I would say mostly ROP is not there. The animal is thrown along with the placenta, where you find focal necrosis of the uterine caruncles and cotyledons. And chlamydia also is a problem because it is very endemic in the case of rams and bucks, and that will be causing infection to the breeding animals, leading to chlamydial abortions. But chlamydial abortion is not usually occurs. Although the animal it is present as a commensal in the animal, chlamydia abortions occur whenever there is a overpowering of whenever if you were able to control the brucella in a particular herd. Even in CRG, we found abortions occurred in certain year where the brucellosis have been totally controlled. We found fewer abortions which are positive for chlamydia. So chlamydia abortions are not very common, but still chlamydia is present as a commensal. Commensal, when there is a immunocompromise, probably this commensal can take an upper hand, leading to an opportunistic capability of producing abortions in the animals. This is the typical, the focal necrosis you find along with the placentitis and the elementary bodies in the trophoblast cells. In the placenta also you'll find elementary bodies. You find focal granuloma or microabscesses in uterus. And we of course have a PCR-based test for detecting the Chlamydophila abortus also. Coming to the Coxiella burnetii. Coxiella burnetii is a very important zoonotic pathogen again, because it's actually causes abortive fever or Q fever. And Coxiella burnetii definitely it's one pathogen to look upon if you are looking at multiple pathogen sets. In CRG, we are trying to develop single 2 PCR for detecting Brucella abortus, chlamydia, Coxiella burnetii, Campylobacter, and also Toxoplasma. All this can be detected by a single tube because each and every pathogen cannot be detected every time. If you do a PCR, just run a single sample, single tube, run the gel, you just find what are all the pathogens that are being involved. Sometimes there can be a mixed infection. Brucella may be there, Coxiella may be there. One may be overpowering the other, but that needs to be looked into pathologically. We need to relate based upon the lesions and based upon the in situ, if we can able to demonstrate which particular pathogen is actually involved in all the pathological picture, that needs to be done. But this kind of studies may not be done at field level. Definitely they are of research interest, but maybe over the period of time to understand how this particular infections and how this particular arena wherein mixed infections are there, how the one is overpowering the other over the other can able to looked upon. So this is very important, that particular perspective. Campylobacter abortions is there. Again, vibryonic abortions are also very, very common. These are microaerophilic pathogens. If we can able to look into, maybe we can try out based upon the clinical history. Of course, Toxoplasma, there's an interesting finding that Toxoplasma can cause abortions at any stage of infection. And Toxoplasma is highly zoonotic in nature. It's very important pathogen, not just sheep and goat, for human beings, for other domestic animals also, where there is a definitely host and intermediate host play is there. Maybe knowing about the bradyzoids and tachyzoids, how that particular cycle is being maintained between the definite host and the intermediate host. And we of course have particular testing, but Toxoplasma has to be dealt very, very carefully because of biosafety level 3 pathogen. in sheep is very important disease, toxoplasma. You will be finding very, very common toxoplasma-induced abortions in sheep, although it's goats, that's less common, but sheep it's very, very common. So that's why I'm saying just abortion means Brucella comes into picture, but you should think about other pathogens also. Just to have an unbiased picture, that's why I'm explaining the other pathogens also. I am explaining little quickly. If someone wants little more detail, I can able to explain detailedly at the Q&A session among this particular. Listeria is very important pathogen of sheep, especially sheep, it causes rattling disease. This particular Listeria, what happens is that this Listeria produces loads and loads because it multiplies very fastly. It produces a kind of bacteremia. It goes and infects the uterus. Loads and loads of bacteria are infecting the uterus, as you can see in this particular slides, were taken from the placenta. You find huge amount of gram-positive bacteria causing this particular pathogenic abortions. In the case of, as you can, like abortion storms, it can occur in the sheep. Again, leptospira is also another important pathogen. All this thing depends upon the history. Like in Gujarat, this kind of leptospirosis is very common. In the case of where people rice field is there, this leptospirosis is very, very common. So certain areas like probably we are found in northern Tamil Nadu, southern Andhra Pradesh, or certain Assam. This kind of disease is very common, where marshy, swampy areas are there. So type of samples that are used for diagnosing abortions, why I put that particular slide is that people usually go for sera. Sera is the most common, most important also, and most widely used sample for detecting various abortion causing agents. But what I want to say is that slowly I think we need to diversify the type of sample that we're sending. Now farmers are thinking like they are sending the blood sample, blood-based PCR for blood samples, and you can send it in the population cards, or we can go for illusion-based techniques, ELISA, as I was saying, and you can collect the genital swabs, like it is preputial swabs, or you can collect the vaginal vulval swabs, or you can also send the fetal material by eyes, dry eyes, where you can go for all the other testing also is possible. So I think we need to diversify the sampling so that you can able to identify not just the antibodies, we can also detect the direct presence of the pathogens. And the type of diagnostic tests are being used like culture, PCR, ELISA. As you can see in Brucella, because you can see the SAT is the most commonly, SAT, RBPT. These are the most commonly used tests, followed by ELISA. The one which are in the brown colored, right? So they are for Brucella. For other pathogen like Coxella burner type, people are going for ELISA. For Chlamydophila abortus, people are going for PCR and immunohistochemistry. So bacterial abortions in small ruminants, as you can see that in the case of Brucella, around 20% definitely it is there. Positivity is definitely it is there. I'm just concluding by saying that the infectious abortions are very important, but I want to say that the non-infectious abortions also need to be clearly taken in mind. So the veterinarian or the clinician should have a completely unbiased critical scenario needs to be taken. You do not, at the moment you look at the abortions, you do not always look into Brucella. Of course, I will say that you need to have a proper gear, like protective gear, like wearing goggles, proper gloves and gumboots need to be worn. But still, I say that you need to have an open mind in observing the abortions. So only then we can able to get the wider picture, the broader picture, so that it will be very useful in a particular area. If the anamnestic history, a clinical picture has been taken, probably you can arrive at a better picture that helps over the years in a micro level, probably in a village level. Although we say India is endemic to Brucella, but the picture varies. We say 0 to 30%. That means some place it is 1%, some place it is 20%, some place it is 15%. Why is it so? That means there is a huge geographical influence is there on the Brucella incidence or Brucella prevalence or other pathogens prevalence. Why other pathogen is overpowering over the other? All these things we will be getting. The zoonotic perspectives are always there. We can always work with the medicos. Probably now medicos are understanding that is very important to work with the veterinarians also. So I think we need to work hand in hand. Probably that is what we call as the One Health perspective. So I think the screening of breeding ewes and does should be done periodically to curtail abortion. There is also one more important thing, because even if you're not keeping for breeding purpose also, if you're keeping the animal for meat purpose also, they may harbor the Brucella. So I say that even these animals also need to be screened, because intromission or the natural service is not the only way the Brucella is being spread. Brucella is being spread by the other means also, that is by feed water. So the ram, bucks, they should be screened every year, at least if you are changing them for breeding for genetic purposes. So these new introduction of ram and pigs to be quarantined, and proper biosafety protocols, biosecurity protocols needs to be followed, and then we need to screen them every year. The conclusions and recommendations, as I was saying, IVRA has developed a new vaccine, and NIAB has come up with a DIVA test. Probably if they are coming into the field, and maybe more new researchers are also developing a lot of DIVA tests. DIVA vaccines, if they come into the field, and that the field studies has to be done exactly to know the perspective, the actual picture of how it is going to control. Probably if you can able to go for a long-term perspective, long-term angle of putting forth all those, various phases of controlling of brucellosis, we put forth, probably you can able to come up with, control of brucellosis in animals. Brucella is one particular thing. All other pathogens also need to be dealt unbiased, rather than just looking into that. And one more thing, serological tests always report a little bit overly, because what I was calling this apparent positivity and true positivity, because based upon the specificity. So because of other febrile reactions, that there may be cross reactivity. So anybody who are doing and interpreting this serological based test needs to be very, very careful before reporting it into national and international forums, or even in seminars, webinars. Probably they will invite some unwanted attention and leading to problem. Because India already we are facing a lot of problem in the global arena in exporting our livestock products. Probably we need to have follow this kind of, we need to have a kind of self control in presenting ourselves before the international community. Thank you, sir.